All right, so we have talked a little bit about the EBI and European databases and all, right? Similarly, we will talk about another very important database, probably the one that we use more often in the practicals, that is NCBI, National Center of Biotechnology Information, NCBI. When it is formed 1988, basically 4th November 4th November 1988. Okay, it's under a part of National Library of Medicine at the National Institute of Health. Okay, NIH is involved with it, and it is there to provide us. It hosts multiple databases under this NCBI, and that can be sequence database for gene, sequence database for proteins, and structural database as well. So we'll see that. What are the aims of NCBI? Okay, so they can ask you a question from that. So what are aims? Let me talk about the aims. Okay, what are the aims? The aim number one is to create and maintain a public database. Okay, to create, let me write, create and maintain a database. Second important aim is to provide a retrieval system. Retrieval system. Retrieval system is needed for retrieving information from the database. A database with so much information will be of no use if you are unable to retrieve the information. So retrieval of system using what? Using entries. Okay, entries is used in the NCBIs to access information and to retrieve information from the database. Okay, and also the third important point is to develop softwares, tools and resources to analyze the data set, analyze the biological data set, and also to help in training, help in computational biology, training in computational biology, to promote the database, data deposition and data exchange, and the standard to database data deposition and exchange. All these are aims of construction of NCBI. Okay, and the NCBI is what? It's a combination of databases, combination of DBs. Okay, and what are some example of such kind of database, the combination? So let me write it here, database this side, and we'll talk about the description. So one of that kind is nucleotide database. The nucleotide database, in this case, this is the sequence database, DNA sequence, RNA sequence database. Second type here, genome database is there. Okay, so where we have complete genome information on complete genome. Then third thing, taxonomy databases. This is for classification of an organism. So classification, animal classification, plant classification database. What else we have? Structural database. And the example of such kind of structural database is molecular modeling database, MMDB, molecular modeling database. And we have experimental 3D structure. Okay, that is the molecular modeling database, where the 3D structure modeling is prepared and the information is stored based on the structure of the primary sequence of the protein. We also have domain databases. Domain of means what? Proteins domain. Okay, and one such example is conserved domain database. CDD. Okay, there is also 3D domain. 3D domain database is also available. Molecular modeling domain database, MMDD or MMDB, molecular modeling database. Okay, and there's one more kind that is known as OMIM. OMIM, online Mendelian inheritance in men. Online Mendelian inheritance in men, that is OMIM database. These are all example of databases that are available, and some more example I'll tell you. SNP, single nucleotide polymorphism database. What else we have? UniSTS, that is sequence tag site. STS means sequence tag site. Sequence tag site markers. Okay. Then we have Unigene. What is Unigene? Gene based expressed sequence. Then we have homologene. Homologene. What is that? Homologene. Eukaryotic homology group. Homology group. The database with eukaryotic homology group. Then there is cancer chromosome databases, where all the chromosomal aberrations related to cancer is only present. Then we have GenSat. What is GenSat? All caps. Gene expression pattern in mouse CNS, where we study gene expression pattern in the mouse central nervous system. Okay. We have GEO database for what? Gene expression data. Okay. We have ProSet. So you can see under NCBI there are even more databases than under EBI. So what is ProSet is for? Population study database. Okay. And finally the one probably you've heard, PubMed. PubMed is for biomedical literature. And there is PMC, PubMed Central, which carries free and full text journal articles. Free and full articles. PubMed Biomedical Literature and PubMed Central or PMC, free full journal articles. And we have some online textbooks in books, and RefSeq is a stable sequence, stable reference sequence for all molecules corresponding to a central dogma, that is RefSeq. So these are all the different kinds. Sometimes they will ask question related to this, 1 marks question or 2 marks question, that what this particular tool is useful, what this database is useful. All you need to understand is that you need to understand whether the term that we are using, whether it's a database or it's a tool. There are difference in that. Okay, database is a different thing, tool is a different thing under the database. Okay, for example, these are all databases we're talking about. We're talking about Geo, we're talking about GenSat, SNP. These are all database where they have the information regarding the biological system, sequence information, structural information, and also storage of information, and access can be done, and we can run the similarity as well. But tools are softwares with which we can access the database. So there is this difference. Okay, it's very important to understand the difference between a database and a tool. A tool is something with which we can gather, we can extract information. We can extract information from database. So for example, what is the example of a tool of NCBI? So these are all database of NCBI, right? So what is the tool of NCBI? So let me write NCBI tool, and what we'll find out about NCBI tool, enters. So whenever you open this NCBI website, you'll see this enters term. which has an integrated information database retrieval system. So basically it's a DB retrieval system, database retrieval system. So basically with enters we can get information out from any database which is under NCBI, which has a very simple user interface with which all the components of different database of NCBI are fed, so that we can access those different information. So this is NCBI, and let's say these are all databases, and this is this user interface where we have buttons and keys. Let's say we click this key, then we get information regarding a DNA. Let's say we click this, then we have a sequence of DNA. We can run that sequence, match with all the database here and get the answer. So like that. Okay. So this user interface is enters for NCBI. Enters is the biggest user interface with which we can access all the databases under NCBI. Okay. One more tool that is available. So let me write. So NCBI, and we're talking about tools, and remember we have discussed about enters. Now second important tool, most used tool is BLAST. Second most used tool, basic local alignment search tool, BLAST, basic local alignment search tool. Okay, a program for sequence similarity, finding sequence similarity against a variety of sequence databases. So we'll find sequence similarity against various sequence databases. Okay, and there are different kinds of BLAST, different sorts of BLAST tools that are available. Let me tell you that. Okay, so the standard kind of BLAST, what else we have? We have Mega BLAST. We have what kind of BLAST?